In our next section, we'll be discussing receptor physiology. In this next section, we'll talk about receptor physiology. As shown previously, receptors are found integrated with a cell membrane, typically on the outer leaflet of a membrane, and that's shown here in molecule A. And this molecule, which is a protein, also has a glycosylated moiety, which increases its specificity for interacting with extracellular molecules. Important points to note about receptor physiology. So first of all receptors are proteins, and receptors are activated by extracellular molecules known as ligands. Now there are two major classes of receptors. There are ionotropic receptors, and these receptors themselves are ion channels, such that when they are bound to their ligand or unbound to their ligand, this will alter whether or not the receptor itself opens and becomes an ion channel. Metabotropic receptors, by contrast, are not ion channels. They activate a metabolic pathway to cause their effects. Now often, activation of a metabotropic receptor will open or close an ion channel, but it's important to recognize that the receptor itself, in these cases, these receptors are not ion channels. The next slide further illustrates the difference between ionotropic and metabotropic receptors. On the left, we have an ionotropic receptor. When it's bound to its signaling molecule, the receptor itself opens to an ion channel, and there's a flow of some ion through that channel/receptor. When that ligand, in this case, is not bound to the receptor, the channel closes and the ion cannot flow through. Shown on the right is a metabotropic receptor. Here, the receptor binds to its signaling molecule or ligand. That's going to activate some intracellular metabolic process. In this case, a G protein will be activated, and that will activate some other intracellular mechanism, which may or may not involve the opening or closing of an ion channel. There are two major classes of signal transduction mechanisms that we'll talk about. There are G protein linked receptors and tyrosine kinase linked receptors. And it's important to remember the end result of what these receptors are actually doing. What they will do is ultimately modify the phosphorylation state of a population of intracellular proteins. And we'll describe this in some more detail as the lecture moves on. We'll begin by talking about G protein linked receptors. As shown in this slide, the G protein linked receptor cascade can be complicated. There are multiple steps from the initial point of activating the receptor shown at the top left, point 1, all the way through until other molecules inside the cell become either phosphorylated or dephosphorylated, and therefore will alter the protein shape and function shown at point 5. The important point to remember is what's actually happening here. We have a first messenger, or an extracellular messenger, some molecule which cannot get into the cell but needs to exert an effect onto that cell, and therefore that molecule needs to induce the production of a second messenger. The second messenger in the case shown here, as is the case for two kinds of G proteins, is cyclic AMP. And we'll talk about in some more detail about the production of cyclic AMP and how G proteins are activated as part of this pathway. We'll begin by talking about the biology of G proteins. G proteins are heterotrimeric GTP binding proteins. Heterotrimeric just means that there are 3 different segments that make up the overall G protein complex, and they're considered to be a molecular switch. That is, depending on whether they're bound to GDP or GTP, this will determine their activity level. At rest, inactive G proteins are bound to GDP. When they are activated by their receptor, and we'll show you how this happens in the next slide, when they're activated by the receptor, they will release GDP, there will be a brief transition period where no nucleotide is bound to the G protein, and then a new molecule of GTP will bind to this G protein to activate it. Activated G proteins will then go on to interact with other molecules, as we'll see soon, and this activation state will last for a temporary period and will end with the hydrolysis of GTP and the loss of inorganic phosphate as shown on the bottom of this slide. G proteins will then interact with adenylate cyclase, and when they interact, they will do this via what's known as their alpha subunit. That subunit and its interaction with adenylate cyclase is shown on this slide. So on the left, we have what's known as a stimulatory receptor. That is, this is a receptor that is bound to a G protein and will ultimately activate adenylyl cyclase. The mechanism by which this happens is shown here. This heterotrimeric G protein has 3 subunits. There's an alpha in blue, a beta in green, and a gamma in gold shown here. When the receptor is activated by its agonist or by a ligand, it's going to release the alpha sub S subunit, and S in this case stands for stimulatory. This alpha sub S subunit will interact with adenylyl cyclase to activate it, and as we'll learn soon, this will cause the production of cyclic AMP from ATP. Alternatively, there are other receptors bound to other kinds of G proteins that will activate their G protein, will release an alpha subunit, but this alpha subunit, instead of activating adenylyl cyclase, will inhibit it. These G proteins are known as inhibitory G proteins, or G sub I standing for inhibitory, and the only difference between these G proteins and the stimulatory G proteins is in their alpha subunit. So the alpha subunit is given its own name, alpha sub I. The major difference then between these two G proteins is how they interact with adenylyl cyclase. It is also worth noting here that you may come across more than one term to refer to this enzyme. Here it is called adenylyl cyclase, although you may hear it said or see it written as adenylate cyclase. Ultimately, the activation of adenylyl cyclase will cause the conversion of ATP to cyclic AMP, shown here, and that cyclic AMP is going to interact with a cyclic AMP-dependent protein kinase, that is, an enzyme that will phosphorylate other proteins, and that phosphorylation step will cause a change in the physiological state of this cell. The specifics of what actually happens will vary cell to cell, depending on the complement of proteins that are expressed in that given cell. You may have noticed that this is a fairly complicated system to transduce an extracellular signal into an intracellular signal. One rationale for having such a complicated system is that it leads to amplification. That is, when a single ligand binds to its receptor, it may ultimately activate many molecules inside of the cell. And that's because at any step along the way, there are multiple products of each individual step. So for example, when a receptor activates with its ligand, it may activate 10 different molecules of adenylyl cyclase. Each of those adenylyl cyclase enzymes may create another 100 cyclic AMP molecules, and so on down the line. This suggests that a single extracellular ligand, when it's bound to its receptor, may cause the activation of 10 million molecules inside of the cell, as each of these steps has its own amplification. One important point to remember about these mechanisms is the physiological control that exists upon this mechanism. So this slide summarizes many of the features that we've already discussed, and that is a stimulatory receptor, when activated, will cause its G protein's active subunit to gain GTP and lose GDP, and then will activate adenylyl or adenylate cyclase to cause the production of cyclic AMP. Or an inhibitory receptor, when it gets activated, will also cause the activation of its G protein, but when it gets activated, it will inhibit adenylyl or adenylate cyclase, which would inhibit the production of cyclic AMP. One important feature not yet discussed is the role of an enzyme known as phosphodiesterase, or PDE, in this process. PDE will shut down the production of cyclic AMP. This is extremely important. If this mechanism did not exist, one would not be able to regulate the production of cyclic AMP. And as we'll learn later, PDE is an important target for many drugs that we'll be learning about. The end product of the action of PDE on cyclic AMP is 5' AMP, which is a similar molecule of cyclic AMP but without the ring. There's a third kind of G protein known as G sub Q. G sub Q has a slightly different mechanism. Shown here as an example is an alpha-1 adrenergic receptor, which is linked to the G sub Q pathway. It's important to remember this could be the case for any G sub Q pathway, not just alpha-1 receptors. But in this case, when the alpha-1 receptor is activated, it's going to activate its G sub Q protein. That G protein is going to get rid of GDP and gain GTP, and in this respect is identical to the previous examples of G sub S and G sub I. Where this mechanism differs from the previous two is that instead of interacting with adenylyl cyclase, this G protein will interact with phospholipase C, or PLC. PLC is an enzyme which will split PIP2, phospholinositol biphosphate, which is a phospholipid within the cell membrane. It will split this molecule into IP3 and DAG. IP3, which is inositol triphosphate, will go into the intracellular solution, or the cytosol, and will activate receptors on the endoplasmic reticulum to cause the release of calcium into the cytosol. That calcium will bind to a calcium binding protein, and that will have some physiological effect, and the specific effects will vary depending on the complement of proteins that are expressed by that given cell. The other product of the PLC reaction is diacylglycerol, or DAG. DAG, along with calcium, will both activate protein kinase C. Protein kinase C is a kinase which is bound to the inner leaflet of a membrane, and when activated will phosphorylate proteins that it interacts with. The specific physiological effects of this activation will vary depending on the complement of proteins expressed by that given cell. Shown in the next slide are all the abbreviations shown from the previous several slides. There are several clinical conditions when G proteins become important. So one clinical condition is cholera. And as referred to here, this is a situation where you are stimulating the stimulator. So the cholera toxin is going to affect the G sub S alpha subunit, and it will prevent alpha sub S from dissociating from adenylate cyclase. It's important to recognize that under normal circumstances, alpha sub S will only interact with adenylate cyclase for some short period of time and then release from it. The alteration of alpha sub S by the cholera toxin will cause this dissociation to be impaired. Therefore, there will be a large increase in cyclic AMP. In the specific cells that are affected by this toxin, that is the GI mucosal cells, an increase in cyclic AMP will increase the activity of a pump, which pumps chloride out of the cell and into the lumen of the gut. Sodium and water will passively follow, and this will lead to a massive osmotic diarrhea. Another example of a pathological condition involving a G protein is pertussis, or whooping cough. In whooping cough, we are inhibiting the inhibitor. That is, the toxin will affect the G sub I, or the inhibitory alpha subunit. It will cause this alpha subunit to more rapidly dissociate from adenylate cyclase. The result of that will be a lack of inhibition of adenylate cyclase, And similar to the previous example, there will be a large increase in cyclic AMP. The previous example, in the case of cholera, the disease locus was found in the GI tract. Here, because of the fact that this is a respiratory toxin, the disease locus will be in the respiratory tract. And respiratory epithelial cells that are ciliated, when they have an increase in cyclic AMP, they no longer have the ability to regularly or appropriately regulate the motion of those cilia. And without that regulation, the only way to get rid of toxins that are inhaled is via forceful or whooping cough. Why else are G proteins important? As we'll discuss later, all autonomic receptors utilize G proteins. Another related form of signaling is that shown here as it relates to nitric oxide. Endothelial cells and smooth muscle cells have to interact in order to allow the vascular system to change its diameter as various stresses are incurred. Guanylate cyclase is a molecule found in smooth muscle cells that will cause the production of cyclic GMP from GTP, and that cyclic GMP will trigger a cyclic GMP protein kinase to cause smooth muscle relaxation. Nitric oxide will activate the soluble guanylate cyclase in order to potentiate this process. It's important to recognize that this is related to a mechanism by which a long-standing class of drugs, known as the nitrates, have been used to treat things like coronary ischemia and angina. These drugs have been used for 30 years without a clear understanding as to what their mechanism of action was, and as we've found out over the past decade or so, their action is likely related to nitric oxide acting on this system. There are also normal physiological mechanisms by which this system is activated. Acetylcholine, histamine, or ATP, released locally during tissue injury, will activate nitric oxide synthase. Nitric oxide synthase will cause the production of nitric oxide through the breakdown of arginine into citrulline and this ultimate nitric oxide. Another important clinical correlation here is the fact that PDE type 5, this is a phosphodiesterase as described earlier, will break down cyclic GMP into 5' GMP. This is the main target of drugs for erectile dysfunction. By blocking PDE5, one will cause more smooth muscle relaxation, and therefore this will treat erectile dysfunction. This also indicates the interaction between drugs like nitrates and drugs for erectile dysfunction and why they are contraindicated to be taken simultaneously. Question 1. To distinguish between hypoparathyroidism and pseudo-hypoparathyroidism, which is renal resistance to parathyroid hormone, one can give an infusion of parathyroid hormone and measure. Hint, parathyroid hormone acts on G sub S. A. Urine IP3 levels. B. Serum albumin levels. C. Urine cyclic AMP levels. Or D. Serum phosphodiesterase activity. The correct answer here is C. Urine cyclic AMP levels. The reason is, as parathyroid hormone acts on G sub S on cells in the kidney, and as these cells are ultimately sloughed off, one can measure the production of cyclic AMP in the urine. This is the most direct measurement that we have of activation of G sub S on renal cells. The other answers here are incorrect for the following reasons. Urine IP3 levels would not be correct because IP3 is not acted upon by the G sub S system. Serum albumin would not have any relationship to G sub S activation, nor would serum phosphodiesterase activity. Question 2. 77-year-old man in the cardiac ICU has decompensated congestive heart failure, and he's in cardiogenic shock. You're trying to give him inotropic support. Increasing cyclic AMP levels will increase cardiac contractility. Which drug would not help in this case? A. Milrinone, which is a phosphodiesterase inhibitor. B. Digoxin, which increases intracellular calcium. C. Dobutamine, which binds the beta-1 receptor and will stimulate G sub S. Or D. Verapamil, which is a calcium channel blocker. The correct answer here is D. Verapamil, the calcium channel blocker. And the reason is the rest of these compounds will ultimately help cardiac contractility. Milrinone, by being a phosphodiesterase inhibitor, will increase cyclic AMP levels, which, as stated in the question, will increase cardiac contractility. Digoxin, by increasing calcium levels inside of cells, will directly increase muscle contraction. And dobutamine, similar to milrinone, by binding the beta-1 receptor and activating G sub S, will increase cyclic AMP levels. Verapamil, by decreasing the influx of calcium, will decrease the point stimulus to increase contraction. Question 3. Joe Footballer is injecting steroids to enhance his performance. Which of the following will block the action of these drugs? Phosphodiesterase inhibitors, calcium antagonists, inhibitors of DNA transcription, or GTP analogs. Correct answer here is inhibitors of DNA transcription. The reason is steroids, because they are nonpolar, can directly pass through membranes and therefore do not have to interact with any of the signal transduction systems that we've been talking about. So phosphodiesterase inhibitors, calcium antagonists, and GTP analogs would certainly have the potential to disrupt activation of the G sub Q pathway in the case of calcium antagonists, or any of the G protein pathways in the cases of phosphodiesterase inhibitors or GTP analogs. They would have no effect on steroids, which don't utilize these mechanisms at all. Tyrosine kinases. So tyrosine kinases are the other major form of signal transduction pathway that are mediated by a membrane receptor. One major difference between tyrosine kinases and G protein link systems is the type of ligand that's involved. Typically, for tyrosine kinases, growth factors are involved. So things like insulin-like growth factor, nerve growth factor, insulin itself, which is a growth factor. These all utilize tyrosine kinase receptors. When a tyrosine kinase receptor is bound to its ligand, and there's another similar receptor also bound to its ligand, these two receptors will dimerize. After dimerization happens, there will be a self-phosphorylation step, such that these molecules will phosphorylate themselves on tyrosine residues, and this will cause the activation of intracellular proteins, because as these tyrosines are phosphorylated, this whole dimer complex becomes its own kinase, and it will go on and phosphorylate other molecules. And as each molecule can phosphorylate multiple other molecules, this also has an amplification factor, as was described previously for G proteins. It's important to remember that the end result of activation of tyrosine kinases is typically the alteration of gene expression. Again, typically it is growth factors that interact with tyrosine kinases, and the end product usually is the activation of a transcription factor to alter the expression of a particular population of genes. Clinically, molecules that inhibit tyrosine kinases have found some popularity in the treatment of cancer. One example of this is Gleevec, or imatinib. This is a small molecule inhibitor of a receptor tyrosine kinase currently used to treat chronic myelogenous leukemia and GI stromal tumor, or GIST. Shown here on the bottom of this slide are a series of FDG PET images which indicate the treatment effects of Gleevec. So shown at baseline is metastatic GI stromal tumor, shown with the dark images around the peritoneal cavity, and with treatment using Gleevec, one can see how inhibiting this receptor tyrosine kinase can potently decrease the activity of these cells. That concludes this section on receptor physiology.